The mystery behind the billions of starfish deaths revealed. Imagine a world where billions of starfish suddenly vanished from the Pacific coast of North America, leaving behind only devastation and mystery. This ecological disaster, which began in 2013, wiped out an estimated 5 billion sea stars stretching from Mexico to Alaska, affecting over 20 different species. But here's where it gets truly fascinating. Scientists believe they've finally cracked the case. Alyssa Gaiman, a marine disease ecologist from the Hakai Institute in British Columbia, Canada, played a crucial role in identifying the culprit. She described how healthy sea stars, also known as starfish, typically have puffy arms extending outward. However, during the wasting disease outbreak, these creatures developed lesions, and their arms would eventually fall off. The sunflower sea star species suffered the most severe impact, with nearly 90% of its population decimated within just 5 years. For over a decade, researchers pursued various leads, but the true cause remained elusive. Some focused on the wrong type of virus, while others examined dead sea stars that no longer contained the vital coulombic fluid surrounding their organs. It wasn't until scientists analyzed this fluid that they made a groundbreaking discovery: the pathogenic bacterium Vibrio pectenicida, which also infects shellfish. This revelation, published in the journal Nature Ecology and Evolution, marks a significant milestone in understanding one of the most pressing marine diseases. Rebecca Vega Thurber, a marine microbiologist from the University of California, Santa Barbara, who was not part of the study, emphasized the importance of this finding, stating that it addresses a long-standing question about a very serious disease in the ocean. The disappearance of sea stars had far-reaching consequences for coastal ecosystems. Without these natural predators, which feed on virtually everything on the seabed, sea urchin populations skyrocketed. In Northern California, this surge led to the destruction of approximately 95% of kelp forests over the past decade. Kelp forests, often referred to as the rainforests of the ocean, are vital underwater habitats that support a diverse array of marine life. Callum Duncan, head of policy and advocacy at the Marine Conservation Society in the UK, highlighted the delicate balance of these ecosystems. He noted that the overgrazing of kelp by sea urchins underscores the importance of maintaining healthy marine environments, which play a crucial role in combating the climate crisis. Scientists are optimistic that their findings could pave the way for sea star conservation efforts. They are exploring options such as relocating the animals or breeding them in captivity before reintroducing them to areas with depleted populations. Additionally, researchers are investigating whether treatments like probiotics could enhance the sea star's immunity to the disease. Meanwhile, governments around the world are convening in Geneva to negotiate a global plastics treaty aimed at preventing plastic pollution from harming marine life, disrupting fishing industries, and affecting tourism. The UK government has called for a bold and ambitious global agreement to eliminate plastic pollution by 2040. Marine Minister Emma Hardy stressed the UK's commitment to promoting an effective treaty that fosters sustainable plastic consumption and production, addresses problematic plastic products, and advances a circular economy. What do you think about the potential impact of this discovery on marine conservation? How might the identification of Vibrio pectenicida influence future research and protection efforts? Share your thoughts in the comments below. Sea star wasting disease. Vibrio pectenicida, FHCF3 Unmasked. The strain FHCF3 of Vibrio pectenicida has been identified as a key contributor to the phenomenon known as sea star wasting disease, which has raised significant concerns among marine biologists and ecologists alike. This disease has led to alarming declines in sea star populations, particularly affecting species like the sunflower sea star, Pycnopodia helianthoides. For those interested in the scientific data surrounding this issue, comprehensive datasets, including metatranscriptomic and 16S rRNA gene sequences, are securely stored in the NCBI Short Read Archive under bioproject number PRJNA115080. Additionally, the complete genome of V. pectenicida strain FHCF3 can be accessed through the NCBI GenBank repository with the accession number JBLZMR0000. Raw sequence reads are also archived in the NCBI Short Read Archive, specifically under bioproject number PRJNA12322168. Furthermore, the full 16S rRNA gene sequences for this strain are available in the NCBI GenBank repository, with several accessions including PQ707178 and PQ76322 through PQ763229. For those seeking source data, it can be found in the associated paper published in Nature. In terms of code availability, the computational code developed during this study is accessible via Dryad at the following link: https://doi.org/10.5061 Dryad.5 MKKWH7G9. The references cited in this research provide a wealth of information on the topic, including studies that explore the mortality rates of ochre stars during the 2014 wasting disease outbreak, the role of environmental factors, and the association of densoviruses with sea star wasting disease. Notably, research has shown that disease epidemics, coupled with marine heat waves, have contributed to the significant decline of pivotal implications of these findings are profound, as they not only highlight the fragility of marine ecosystems, but also raise questions about the future of these species in the face of climate change and disease. As we delve deeper into the complexities of marine health, it becomes increasingly clear that understanding the interactions between pathogens, environmental conditions, and host species is crucial. What are your thoughts on the impact of climate change on marine biodiversity? Do you believe that current conservation efforts are sufficient to combat these challenges? We invite you to share your opinions and engage in this critical conversation.